Thank you very much. It's a real pleasure to be back in Santa Barbara. What a beautiful place you live in, you lucky, lucky people. Um, so our Max Planck Institute has existed since the 1st of June. So it's one of the newest, it's definitely the newest Max Planck Institute in the society. And so I've always been fascinated by collective behavior. The sorts of footage you see like this of schooling fish on a coral reef or flocking birds. And what's perhaps surprising is despite the ubiquity of these behaviors, we still only have a very rudimentary understanding of both how and why organisms like this coordinate their behavior. Yet collective behavior is a fundamental principle of life on the planet, and that's what we're very interested in our department. And so we also study the simplest animal on the planet. This is called Placozoa. It's a basal metazoan. It's in its own phylum. And if you track the nuclei of this organism, the density of the nuclei, and the direction of the nuclei, or the direction of the motion of the cells, you can see again these types of hallmarks of local interactions scaling to large-scale collective properties. We also, as Jeff mentioned, we've also been working a lot on locusts. So these are very important insects because they impact the livelihood of 1 in 10 people on the planet. Yet nobody understood how and why they behave like this. It looks cooperative, but in actual fact, conducting a series of experiments, creating this sort of particle accelerator for locusts, not the smartest animals out there, they'll think they're in this never-ending locust swarm. We discovered that in fact these swarms are driven by cannibalism, cannibalistic interactions. Every individual is trying to escape being eaten by the one behind and trying to attack those ahead. So it's when they run short of essential nutrients, protein and salt, that they form the swarm. So we have to be very careful when we see coordinated behavior, not to confuse that with cooperative behavior. And so we need to think about the selection pressures at work. Now, that video I showed you was the state of the art in the sort of mid-2000s, but at the same level of resolution now, using deep learning, we've been able to develop software that can track the full body posture of locusts and can do so for thousands of individuals simultaneously and in real time on standard computer software using GPU acceleration. And so really there's a new age of studying behavior. We can then use dimensionality reduction techniques to really try to understand what do we mean by behavior. And this type of software is so robust. Later on I'll show you it with zebra, but you can also apply this to Drosophila, for example, a very common model organism in the biological sciences. And again, you can get great detail regarding behavior. So there's a new era, as I mentioned. You know, we're really able to do things that we could only dream of only a few years ago. And we can do this for many, many animals simultaneously to really understand collective dynamics. Again, these are locusts, sped up now, but you can see the detail. Now, arguably, the most complex animal we study are humans. And this is in a main railway station in London. And unbeknown to these pedestrians, we have actors within the crowd subtly manipulating subconscious aspects of their behavior, in this case, joint attention. But this also highlights, again, the fact that we've developed software that tracks the motion of the pedestrians, but also, in this case, records or estimates where they're looking, so we can understand the information processing within this natural social context. So information transfer is really the hallmark of collective behavior in the biological world, whether you're looking at neural collectives or organismal collectives. And this is what differentiates these systems from physical collectives, for example. And so you can see why in the 1950s and even into the 1960s, it was believed there must be telepathy or thought transference among the individuals. But what we now know, this is a computer simulation that I've developed, what we now know is that relatively local interactions among the components of the system are sufficient to explain many of the properties we see. This is called self-organization. There's no global orchestrating figure telling the others what to do. And so here we can pause and we will rotate around. You can see the vacuole of the individual here. And so this has been a very useful approach to try to understand collectives. And why is this so fascinating? Well, I mentioned it's ubiquitous, but also neurobiologists are focused to a large degree on how does the individual mind work. And of course, that's absolutely critical. But what I've become fascinated in is how does social connectivity connect minds together? Can we think of collective minds or collective cognition within animal groups? And if so, what sort of flavor does this type of interacting minds give rise to? What sort of cognition is special here? How does this complement our understanding of individual level cognition? And so because very few of us are studying these types of principles, I'm really only going to give you, scratching the surface of our understanding. And when you look at this intricate interplay of interactions, you can see why we cannot just use verbal argument alone to try to explain how these local interactions scale to collective behaviors. So as I mentioned before, we've resorted to computer simulations to try to get inside the head of this individual. But models, and I'll show you models throughout my talk, are incredibly important for structuring our thoughts about collectives. But ultimately, we really need to test these ideas using real experimental systems. So when I was able to go and set up my lab when I was a professor at Princeton from 2008, I got very excited about working with these organisms. These are golden shiner fish because they're a vertebrate, so they have to solve the same problems that many other vertebrates have to solve. They have to avoid risks, they have to find food, they're living in a dangerous, unpredictable, time-varying environment. But they're also relatively convenient. Like my group is mostly made of physicists and mathematicians and biologists, a few, but we're not very good at catching fish. It was quite hilarious out in Lake Carnegie catching fish. And when we were there, I noticed the fishermen were hunting with bait, and in their buckets, they had this really strongly schooling fish. And I was like, where did you get these? Oh, you can just order them online. You can get 1,000 of these delivered to your door for $70, including shipping. And so this solved our problem. And you can see they're beautiful organisms, and they exhibit these rich dynamical properties. We also got lucky because even if you give them deep water, they'll swim at the surface, so it's a quasi-planar situation. And as you'll see later, they rely almost exclusively on vision, as opposed to sensing hydrodynamic cues, which makes it easier for us to get at the sensory system. So here you can see the group undergoing these wonderful transitions in behavior. And as I mentioned before, with the advent of deep learning in particular, but even before that, we've been able to develop software that can track the motion of all of the individuals simultaneously and even maintain the identities based on pattern differences on the surface of the fish that to a human are indistinguishable, but to a deep neural network, it can specialize on this particular task and become very good at recognizing who is whom. And so I mentioned to you predation before, and I'll come back to the role of predation in selecting these types of group behaviors. But I want to show you another example of information processing in these collectives. And this is work that I did with Andrew Badal when he was a PhD student with me, and Colin Toney, who was a postdoc. Andrew is now a professor at the University of Washington, and Colin, a senior lecturer at Glasgow University. And so what we did in these experiments was we set our fish a computational challenge. Now, this could have been olfactory gradients or whatever, but we ended up using light gradients because it's much easier to work with. The fish have a natural instinct to want to be in the dark regions. That's where they feel safe from predators, especially airborne predators. But because we can't see them in the dark regions, of course, that's why they want to be there, what you're seeing below is exactly the same experiment as above, but we're now cutting out the visible light, and we're filming them just in infrared light. And then what we're doing, we're projecting onto the base of a tank a time-varying dappled light environment where there's always one globally optimal solution, but also local suboptimal peaks. So the challenge here is that this light gradient is going to be changing, and the challenge for the fish is to find the safest regions. And so here you can see this projected light regime, and we, of course, can control the length scale, the noise, and so forth. We then can isolate the fish using the infrared light view, where we cut out the visible light. But because we're projecting this environment, we can put that back in computationally to then ask, how do the individuals interact with each other? And also, how do they then interact with this complex environmental gradient? And indeed, we were able to find the first experimental evidence that as group size increases, the individuals become much better able to solve this problem. Okay? So for small group sizes, for one individual or two individuals, you can see that they're only about 10 or 15% better than completely random motion, but they get much, much better as group size gets larger. So we thought to ourselves, this is wonderful. We've got these tracking data, so we could look at the accelerations that the individuals exert to get a sense of what they're trying to do. Over what range are they detecting the environmental gradient? And how are they responding to each other? Because we were thinking that they're solving this problem in the same way as is called the wisdom of crowds. So in the wisdom of crowds, this is a concept actually invented by Sir Francis Galton, a cousin of Charles Darwin, who once went to a livestock fair. He was a polymath. He knew lots of things, but he didn't know much about livestock. And there was a competition to guess the weight of an ox. And he realized almost 800 villagers had already entered the competition. So he used their estimates, calculated the mean, and he was 1 pound off the true weight of the ox. So the idea is if you've got many imperfect estimates, if you pool them together, then you can achieve collective intelligence. Now, of course, our fish, to our knowledge, can't do calculations like this, but the social interactions might be pooling the information effectively. That's what we thought was going on, but one of the great things about doing real experiments rather than just theory is that you get surprises. Because while the blue line here shows this very strong importance of the social vector, the role of the social interactions, we found no evidence, regardless of group size, whether you're a group of 2 or a group of 256, that you detect the environmental gradient over any length scale. They are not integrating in time, they are not integrating in space. So this leads us to a conundrum. If the individuals are not utilizing the light gradient at all, how on earth is the group able to detect the gradient so effectively? Natural selection turned out to have found an elegant and cognitively minimalist solution. So as many of you in the room know, the brain is a very expensive organ to run. And so what the fish are doing is they are just measuring the local scalar measure of how good the environment is where they are now. If the environment is not good, if it's light in this example, they will move fast. If they like it, they will slow down. A very simple rule. But that does not have any gradient sensing or taxis. This is called kinesis. And it gives them that 10 to 15% advantage over random motion. But it can't give them that huge advantage you get as a function of the group. Where does that come from? Well, that comes from the interactions. The interactions, that social network of connectivity, facilitates an awareness of the environment that does not exist at the individual level. It's a truly emergent property. And so one way of thinking about this is if my hands are 2 fish, and imagine they're connected by an elastic band or a spring, right? And they're moving together. And then this one reaches a dark region and it slows down. It's almost like there's an increased friction to it. This one doesn't know that, so it continues moving fast. That will start exerting a stretch to that elastic, which will exert a centrifugal force towards the dark region, even though the individual doesn't know it's there. And like cars on a highway, when you're moving slowly, because time to collision is the important thing, you become more densely populated on the highway when you're moving slowly. The same with the fish. And so, on average, probabilistically, the attraction vectors will point towards these dense regions. Another way of thinking about it, and if you take the model to the hydrodynamic limit, you can show this is the case, is that they're basically undergoing a phase-like transition. They're solidifying when they're moving slowly, they're liquefying when they're moving fast, and that is what causes the computation. And we've been able to show this also in evolutionary models. So purely selfish, genetically selfish individuals will readily evolve to have this physical intelligence. And our golden shiner fish have inspired shiner bots. little robots that don't need to be smart, they just need to communicate with each other and interact together, much like their famous killer bots at Harvard. And so I used this analogy of a spring or an elastic between my hands to represent the social interactions. And to a first approximation, this type of Leonard-Jones type potential is okay to explain the behaviors. But of course, these are not individuals employing social forces. If you look at the modeling of collective behavior, 99.9% of the models assume social forces because this comes from chemistry and physics. In reality, of course, they're using visual cues or sensory cues. In our case, our species uses vision. And so we've also developed software that tracks not just the positions, but estimates the body posture, estimates where the eyes are, and then uses graphics processing units to reconstruct how the pathways of photons fall onto the retina for each individual, so we can see the world from the perspective of the animal itself. So for this focal individual, we can see its left visual field in red and its right visual field in blue. And immediately, if you think of the world from its perspective, this is going to be a complex, dynamic, time-varying environment, due both to the motion of others and to self-motion. So you can see why things like social forces are not going to be a good approximation to this type of dynamic topology of communication. And indeed, we've been able to show that this is a much better way to study these systems. So one of the hallmarks of collective behavior is that the change of behavior of one or multiple individuals can change the behavior of others and change the behavior of others. Again, you see waves of activity in neural networks. You see waves of behavioral change within these social networks. And this is work I did with Bryn Rosenthal and Colin Toomey, who are PhD students in my lab. And so what you're seeing here is a slow motion video of a startle response. And this evasion behavior, it's called a C-start in fish, or startle behavior in general is very, very common. You've probably all seen birds and fish exhibiting these very fast waves. Remember, this is slowed down. But the problem, if I wave at the fish, or if I drop something in the water, or if I show them a predator, then I don't know when I look at their response whether it's due to a social response to others or a direct response to that stimulus. As Max Weber said, when people put up their umbrellas together, is it because they're copying each other or because it's raining? So we need to decouple this. So what we did is we developed a little fiber system that will go up and tickle the belly of a fish. And 100% of the time, this will startle them. And the startle is driven by two giant neurons, predominantly, called Meissner cells. So it's a reflex. And if we miss just by a millimeter or so, there's no response. Okay, so we can then know exactly who we've triggered, where they are in the group, and we can then see this cascading behavioral response. But if we do this hundreds of times, so we did this 205 times, where we know the initiator, and we then look at the cascade size, so this is how this avalanche of changing behavior on a log axis versus the log number of events, you can see that this is a very long-tailed distribution. Most of the time, in fact, nothing happens, where only one or two individuals change behavior. But sometimes we get these large cascades that change the behavior of the whole group. And a priori, it's hard to understand why. We're always doing the same thing. There's always one individual changing behavior. Yet sometimes it doesn't do anything, and sometimes it does something massive. Now, this type of long-tailed distribution is also characteristic of things like news spreading on Twitter or Facebook. Which stories go viral or not is very, very hard to predict. Most stories don't, but some can spread very broadly. Or power outages. And so to understand what's going on here, we had to establish the functional mapping between visual input, remember our fish are exclusively visual, we've been able to check this, and motor output. And so if we look at the visual input for a moment, and this is true of any sensory modality, by the way, what the brain has evolved to do is to take in this highly complex sensory information. So this is us reconstructing from the left eye and the right eye how the world looks. And the brain has to take this information and process it and make a very low-dimensional behavioral output. In our case, just 2 neurons. So it's called dimensionality reduction, to take this complexity and reduce it down. And I won't go through the details of this. We can see it in our published paper. But we really needed to do this computational visual field reconstruction. We needed to see the world from their perspective and the body posture mapping. And then using some new advances in machine learning, we're able to work out which visual features do they pay attention to when they are translating this complex scene into their escape behavior. And for those of you interested, I can talk about this later, but suffice to say, it turns out that they're the 2 simplest visual features they could have used. Okay? Again, likely for redundancy and resilience. And so now, we reconstructed the visual field, we've worked out what they're paying attention to, how they translate that into behavior. And so we can now reconstruct the hidden network of communication that gives rise to this collective behavior. Again, if you're a neuroscientist, you can go into the brain to dissect the circuits that give rise to individual behavior. Here, we're effectively doing the same thing, but the social circuits that give rise to collective behavior have no physical form. So we needed to use this new technology to reveal what was hidden to us in the past. But important to note is that even though they're using the simplest visual features they could use, if we zoom in on this network, we can see it's very complex. It's both weighted. The stronger the weight is, the more strongly you're influenced and influence others. It's also directed. So I might be very strongly influenced by you, but you might not be influenced by me, or not so strongly, right? So we have this complex network where the edges here, the weights here, actually represent probabilities, quantitative probabilities. Okay, so what's the probability if you change behavior that I will, and vice versa? And if we look at these networks over time, they're very beautiful. And they're not in the same class. They're not a small world network. They're not a scale-free network. They're in their own unique class. And they have very, very interesting topological properties, such as to minimize correlation of information and to also minimize the amount of time it takes for information to flow through the group. Because these fish have evolved over millions of years under extremely strong selection pressure to process information collectively. 93% mortality per year through predation, for example. So we can ask something very, very basic and fundamental. What is the relationship between the social network structure and social influence? So if we think about this for a moment, it is very often assumed that the most socially influential individuals will be those with many connections to others. In the network literature, this is called a high degree. It just means you've got lots of connections, lots of friends. Okay? And whose neighbors have a low cliquishness. In the network literature, this is called a low clustering coefficient. Well, why is this assumed? It's assumed because people think of behavior spread as being like a fast-spreading disease. And modeling and understanding disease spread has been prevalent in biology for decades, possibly even centuries. Right? And this makes sense in the context of a disease. So let's say I'm infectious. I don't believe I am, but let's say I'm infectious and none of you are resistant to my infection. Then the more of you I interact with, the more connections to others, the higher is the probability that I will infect you. But if I just, for example, infect the front row here, and they're a clique, they don't want to interact with riffraff, the rest of you. They just want to interact with each other. If I infect them and they're a clique, yes, it may spread through this clique very effectively, but if there's no connections outside the clique, then it's not going to spread broadly and lead to an epidemic. So this makes intuitive sense, and people have just assumed that this is the case. But there's no experimental evidence for this. So what we wanted to do is see if we can provide the first experimental evidence that this intuition is indeed correct. So we can ask from the data, not simulating propagation of behavior, actually measuring it quantitatively. Is this true? Well, no. It turns out that the most socially influential individuals, or so-called super spreaders of behavior, are those with a low degree and whose neighbors have a high clustering coefficient. Exactly the opposite of intuition. Again, highlighting the importance of asking fundamental questions, even just not assuming, even though it's incredibly intuitive, even though you think it has to be that way, test it. It's not always the case. And of course, we can then ask ourselves why. And it turns out to be because disease contagion is a so-called simple contagious process, whereas social contagion is what's called a complex contagion. Well, what do I mean by that? So let's assume again that I have a disease. The longer I am in contact with you today, the higher is the probability, and it's only a probability, I'm not definitely going to infect you, but the more I spend in contact with you, the higher is the probability that I will infect you. Now let's say I don't have a disease, but I have an opinion, say a political opinion. I might try and convince you all day of my political opinion, but you may not be convinced. It's certainly unlikely that I will have this increasing probability of then convincing you of my opinion, say regarding your president. Maybe he's too polarizing. But, okay, he's definitely too polarizing for this next analogy, which is, maybe I convince you a little bit. You don't reach threshold, so I convince you a bit, but you haven't changed your behavior. But then I convince you, and then convince you, and then the two of you then talk later, and you're convinced. I've influenced you both directly and indirectly through this loop. And so what we find, whether we look at fish or humans or birds, is that all organisms are seeking reinforcement from multiple opinions before they change behavior. Furthermore, just like humans, we seek information from what seem to be uncorrelated sources. So I remember when I was at Princeton, I went to New York City, and my friends were trying to convince me to buy an album. I love music, but I wasn't convinced. And then I get back on New Jersey Transit to go back to Princeton, and the couple sitting in front of me were talking positively about the same album, and I bought it. My brain sort of saw that as uncorrelated confirmation of the quality, right? So humans do this all the time, but how do fish do it? Well, they do it by passing out the information. They're coincidence detectors, basically, passing out the information in the visual field. If it comes from the same part of the visual field, it may be correlated. If it comes at a similar timing but from a different part, it's likely to be less correlated. But the outcome in terms of the relationship between network structure and contagion is the same. And so it really does change our predictions. And so now what we can do is we can run a whole new set of experiments, and we freeze time when we trigger an individual to startle. We reconstruct the visual fields. We pass this through the algorithm. We know how they translate visual fields to probabilities of changing behavior. We look at the network structure, and we then use our new understanding of the relationship to predict who's going to change behavior when and how far is it going to spread. And now suddenly this real biological complex stochastic system becomes highly, highly predictable. Okay, so what was unpredictable before is now highly predictable. But it turns out that this is really hard to do, this type of work. Really hard. Why is it hard? It's hard because of causality. So if an individual startles, it's an unlikely thing to happen, and then another individual startles shortly after, we can reasonably infer that that second individual startled because of the first one. But what if a third individual startles? Did it startle in response to that second one? Or was it responding to the first one with a greater time lag? Or some balance between the two? What about another one? What about another one? It becomes extremely difficult because we cannot control causality here. And causal inference methods turn out not to work well for these systems. We still don't know why. So we had the idea of creating immersive holographic virtual reality. So we can allow our individuals to interact with photorealistic holograms in the tank with them, interacting with them in real time. So this sounds a bit science fiction-y, but it's based on a very old illusion called an anamorphic illusion. It's so strong that I can tell you right now that this is not a 3D object, but your brains believe it to be until the illusion is broken. And even though it gets put back not quite in the right orientation, it pops back into your brain as being 3D because of the expectations that the world is three-dimensional. Again, this shoe is not three-dimensional. Another way of looking at this illusion is to look at how a three-dimensional scene normally looks on a computer screen, and here one where you're tracking the camera and rendering it from the perspective of the camera. You can now move in behind those objects. They're in the volume with you. So this is the illusion we use. And so we have a virtual fish and a real fish, and 100 frames per second, we update the scene. So we can put one individual into this bowl. We then bounce projected light onto the surface. We correct for distortion. And we can basically simulate anything in this environment using a video game engine. So this is a zebrafish swimming around. This pillar looks really weird to us. But from the perspective of that fish, it looks 3D. It's the same illusion you saw with the tape and with the shoe. So from its perspective, this looks like it's physically there in the tank with it. And of course, the fish will avoid it, even though it's just light. It's not really there. But kind of more interestingly, from my perspective, I mean, you can use this for lots of different scientific studies, but we created a photorealistic virtual zebrafish that swims just like a real zebrafish. Oops. Swims just like a real zebrafish. And here, the virtual fish is just exhibiting a circular motion. But the real fish, you can see, is interacting with it in the full three-dimensional volume. It believes it to be in the tank with it, even though, of course, the projection surface is only the surface of the bowl. But it fully believes that to be another organism in the tank with it. So this is why we call this a holographic projection system. Now, the problem with it is that if I put another real fish into this environment, the world will look distorted to it. The illusion can only work from the perspective of one individual. We can put in as many virtual individuals as we want, of course. But how do we get real individuals to interact? Well, we built 6 of these systems, and we then networked them together such that this individual will interact with a hologram of this one, and vice versa. So now we can have individuals interacting. These 2 individuals are interacting not in the same physical world, but in the same holographic world. And here are 4 individuals. We call this the matrix for obvious reasons. They're interacting in the same holographic environment. Now, this has many advantages to us, because, of course, we can put in virtual organisms. This environment could look naturalistic, or could look infinite, whatever. Put predators, or whatever you want. So, I mean, here we have a very high degree of control over the visual inputs. These fish, again, they're very small. It's exclusively vision at this age in their lives. So we can present the same stimulus on the same part of the retina to the same or to multiple individuals. It allows us to gain much deeper insights into how the brain makes movement decisions. And that's already shown us that all of our models are wrong, but I don't have time to talk about that today. And inspired by neuroscience, we can also adopt a dynamical social patch clamp type paradigm. So the dynamical patch clamp in neuroscience has been used, I guess, for more than 20 years, where you have your algorithm in the circuit with the neural circuit. Right? You're providing the electricity, or you're recording from it. So here now we can test our hypotheses dynamically in the context of the real social circuit. And of course, we can control causality. We can introduce time lags, spatial constraints, and so on. So we're going to hopefully publish some work this year on this. It's pretty exciting. Okay, the last part of the talk, I want to talk about leadership and collective decision making. How do groups of animals make informed, unanimous decisions? I'm going to start with some pretty old work first, because it sets the scene for some very, very recent experimental tests. And I'm going to show you pictures of fish because they're pretty and I like them, but you can think of birds. I'm later going to show you tests with primates. And so you can think very broadly about what this means. And so here, we assume that some individuals in the group have goal-oriented behavior. This could be a cell that's aware of a chemical gradient that the others are not aware of, or an individual organism that knows there's food over there that others don't know about, or perhaps it knows there's a predator over here that the others don't know about, and wants to move away. The model is very, very general. But does this individual with information need to signal to others, I know what's going on, follow me? Do others need to recognize who has information and who does not? Well, we have no experimental evidence for either of these processes. Of course, we could add these things in, but we've got no evidence. So we need to understand, can information be conveyed without signaling and without individual recognition? And so what we assume is that informed individuals reconcile their social tendency with their goal-oriented tendency with a simple continuous weighting term called omega. If omega is small, then they don't want to risk leaving the group. The goal-oriented vector is small. If they really are determined to go that way, this vector can be very important and, of course, could even lead them to lose the group. Now, importantly, I'm going to color in the individuals in bright colors so we can see their informational status. But that's only for us. Individuals do not know the informational status of others. The colors are simply for us humans to see what's going on. They start at random positions, random orientations. There's no cheating going on. And what I'll show you is if informed individuals have a preferred direction, and there's 1, 5, or 10 within the group, in a group of 100 in this case, the green cross just shows you the center of the group. You can see one informed individual is not able to guide the group in its preferred direction. But if we look at 5 informed individuals, after an initial period of reorganization where they occupy the frontal periphery, they sort of guide the group in the right direction. It's not very accurate, but they're kind of going to the right, aren't they? But with 10 informed individuals, suddenly and nearly spontaneously, it's really very accurate. So we can quantify the accuracy of information transfer. 0 means completely random. 1 means perfect information transfer. And we also, by the way, simulate the randomness in individuals' brains. They're not able to perfectly make decisions. And what we see for all group sizes is as we increase the proportion of informed individuals, we increase very steeply the information transfer, and then it flattens out, it plateaus. But if you look at, say, 85% accuracy of information transfer, for a small group like a group of 10, we need around half of them to have this information. But when we get to larger groups, like a group of 200, now we need less than 5% of them to have information. If we simulate cell sheets or migrating herds, this can be an infinitesimal proportion. Yet to the biologist, it looks like everybody knows where to go. But in many cases, acquiring information or detecting the Earth's magnetic field, if you're migrating, can be costly. And so we can expect, and we've done evolutionary models, we can expect there to be a small proportion of individuals that actually hold the knowledge. And I'll come back to that later. But we can test these ideas with our schooling fish. This is not a simulation anymore. These are real fish, where the red are trained to a green laser stimulus and the blue don't have any response. And you saw the wave of information transfer. And exactly as we predicted, as the number of informed individuals increases, it increases the accuracy steeply at first, and then it plateaus. But so far, all of our informed individuals have been in agreement about where to go. But in many groups, they exist in sort of fission-fusion dynamics. And so there can often be disagreements about where to go. And remember, these are selfish individuals. So can they come to a collective consensus? They can't count. I mean, if we were going to decide to go to the same pub together and say 6 of you put your hands up for one option, 5 for another, I could ask the question, do the calculations, pass back the answer. But these organisms, to our knowledge, cannot count. So it turns out what's important are the number of individuals with different preferences, the strength of those preferences, which I'll come back to in a moment, but also the geometry of the problem, i.e. the angle at which they disagree. So I'm going to keep one group to always want to go along 0 degrees along the x-axis. That's represented by the lower dotted line. And then I'm going to assume that the other group disagrees, maybe by a small amount, like 1 or 2 degrees, maybe by a larger amount, like 100 degrees, all the way up to 180 degrees, where they want to go in exactly opposite directions. And I'm going to start the group with random positions, random orientations, and run thousands of simulations and ask, where does the group go? And in this example, there's a group of 100, with 5 that want to go one way and 5 that want to go the other way. And what we find is up to a certain angle, a critical angle, they will split the difference and go the average, up to around 123 degrees. Which, by the way, exactly the angle that the monkey brain also goes from averaging to consensus. And this does not represent the group splitting. What is happening here is just that half of the time above that critical angle, one group wins and they all go this way, and half of the time the other group wins and they all go this way. Now, if you imagine them moving through real physical space towards a red and a green target, if you're far away, the angle is going to be small, so you'll be down here, you'll average, you go down the middle, but you'll always reach that critical point and then make a decision. But what if I add one extra individual to one of the informed subgroups? So now we have 6 versus 5. Random positions, random orientations, no ability to count, and yet now, below that critical angle, they do exactly the same, they split the difference, but now as soon as they get closer and they reach that critical angle, they will almost always, 98% of the time, choose the majority, even though no individual knows that they are in a majority or not. They don't know the information of others at all. And here you can see an equal number preferring the white and the red, and the green ones are uninformed, so it's going to be random. In this case, they choose the red. If I put in one extra white individual, you can see the local noisy nature of the interactions, yet they will almost always choose the majority. And there's a regime where if the weighting of their preferences, if they're really unwilling to give up on their preferences, they will enter a regime where they can no longer reach consensus and they will tend to split. And of course, the green ones may learn about the red and become informed, and so on. Now, when I published this, I was so rushed by the editors, that I sort of saw it as, isn't it amazing that they can do this despite all of these uninformed individuals? It was only years later I realized that the only reason they're able to do this computation is really because of the uninformed individuals. And so later I started looking at this again because there are many examples where there are conflicting interests. If you're in a hiring committee and you can never agree on a candidate, this is bad for the organization. If you're in a fish school or a bird flock and you can never agree regarding which direction to go, you're either never going to find food or be taken out by predators. But what's going to stop a minority from just becoming more strongly opinionated? Can they get their own way? Well, if all individuals have a preference or an opinion, and here we have a majority of 6 versus a minority of 5, but the exact numbers don't matter, and the majority preference is 0.3. As I showed you before, if the minority has the same strength of preference, the majority will win. But if the minority becomes more and more strongly opinionated, they can begin to influence the behavior of the group. 0.5 means half of the time the majority wins, and half of the time the minority wins. But if the minority becomes sufficiently strongly opinionated, they can indeed dictate the behavior of the group, albeit at the cost of much more splitting. But what about uninformed individuals? Because many times, these groups consist of individuals that don't know what's going on, or who are perfectly well informed but don't care whether they go this way or this way. Now, these types of individuals are always assumed to just be a source of noise. So they're just going to mess things up a bit but not change anything. That's fundamentally incorrect. They're profoundly important. So here, the minority has a stronger preference than the majority, 0.38 versus 0.3, but it's not so strong. You can see with no uninformed individuals, the majority is still winning 60% of the time. But if I put in 12 or 15 dumb individuals into the group, this pushes control to the majority. Now, of course, as everybody's uninformed, you can no longer make good decisions. But I can increase the minority preference further, and further still. Now the minority are so strongly opinionated, they win around 98% of the time. But if I put in 12 or 15 individuals that do not care, or are uninformed, or are unbiased, they return control to the majority. Now, we've been able to prove this result analytically in a range of different systems. So we've worked with coupled oscillator systems, we've worked with physical spin systems, so how magnets tend to operate, and most recently in a spin system embedded in geometrical space. And we get the same results again and again and again. A small proportion of unbiased individuals increases the probability of reaching consensus. They allow the two opposing groups to sort of see eye to eye. They make consensus much, much faster, way faster, and much more sensitive. So when I think about neural circuits, you could have each neuron being very good, for example, at detecting which way the world is moving, and maybe being able to come to a good decision. Or you can just let them be noisy and add around 12 or 15% of completely untuned neurons into the circuit, and you should recover the same dynamics as if the circuit was perfect at doing what it does. Right? And this is similar to, but different to stochastic resonance. So there's lots of interesting properties here. But I'm a biologist. I want to test it with real animals. And so the uninformed or unbiased individuals should inhibit the influence of a strongly opinionated minority and return control to the majority. And so returning to our golden shiner fish with postdoc Christos Wanu, who's now a lecturer at Bristol University, who was blind to the predictions of the theory, by the way, when conducting the experiments, we can train fish to have a strong or a weak preference to different colors. So we can have our strongly preferred individuals to yellow, for example, and weak to blue. And the theory predicts that a small absolute number of untrained, unbiased individuals should completely reverse the decision of the group. And indeed, what we find is when there are no uninformed individuals, the strongly opinionated minority wins. But if we put in 5 or 10 untrained individuals, we reverse this property. And we understand a lot about the dynamical principles at work here. But I don't have time to talk about that. But this diversity of opinion strengths, so especially having individuals in the circuit with low opinion strengths that are still participating in the decision, but they don't care the outcome, dramatically changes. And I'm not saying this is a good thing or a bad thing, but I'm just reporting the dynamics. Okay, so we could test this again with a highly complex society of baboons. And this was work with Ari Strangberg-Peshkin when she was my PhD student, who was the first author, and with Damien Farine and Meg Crowford, who's moving to the Max Planck as our third director, the new Max Planck Institute of Animal Behavior. She's a primatologist at UC Davis currently. So we could put GPS collars and accelerometers on almost all adults within a wild, unhabituated baboon troop in Kenya. For obvious ethical reasons, we did not put collars on juveniles. And all the collars removed themselves with a little explosion on a specific date. That's a pretty cool video to watch, actually. And every second of their lives now, we know who's whom. We have the accelerometry information. We have the locations of these wild baboons. And looking at these data, we found tens of thousands of events where there's directional conflicts, where some individuals want to go one way, some want to go another way, and yet, like the fish, they will almost always come to consensus regarding where to go. And don't believe what you see on television. Social dominance, even though there's a linear dominance hierarchy and all males are dominant to all females, it doesn't matter. Subordinate female goes this way, dominant male goes this way, it's 50-50. Right? That could be because there's uninformed individuals, democratizing the process, of course. And so remember these bifurcation diagrams I showed you before? Well, 10 years later, we were able to actually experimentally quantify this within a real system. Now, this model, of course, doesn't capture all of the behavior of baboons, but this phenomenon is so robust, we find it in spin system models and neural dynamical models, in animal behavior. So we actually think this is also how the brain makes decisions. We're doing some really cool experiments with Drosophila now, where we reconstruct the same bifurcations within the brain and with zebrafish. But these baboons are not in an empty tank. This looks like a photo, but this is actually a three-dimensional point cloud collected by a drone. That's me and Ari and Damien in Kenya. And so this clever Swiss technology can image down to three-dimensional accuracy, to 3 centimeters spatial accuracy in 3D, the vegetation of the environment. Their new drone can do 1.5 centimeters accuracy. It looks a bit weird because this is in infrared, so we can get the vegetation index to look at the quality of the resources in this environment. You can even see the luggers and the single tire tracks in this road. You can see the animal trails. It's wonderful data. And so this allows us to put the high-resolution GPS data of the organisms, the baboons, back into the real physical context in which they're moving. So we can take this into account. And Ari, using Bayesian methodology, could formally now start putting complexity into these individuals. They're not particles interacting with forces. They can see each other within this complex environment. They're influenced by the habitat, the resistance of the habitat. They remember where they've been. That could be important too. They also have certain desires, like to get to the sleeping grove or to get to certain food sources. We can put this back in a formal way. And one surprise was they're not really interacting with positions in space, but with a sort of decaying trail of where others have been, possibly olfactory, a bit like ants laying chemical trails. They seem to smell where others have gone in the recent past. And this has given us, by taking into account not just the habitat and the social behavior, but both on this nonlinear axis, gives us much better predictive capabilities in understanding where they move. But you can't always put collars on animals. This is work with Blair Costello, Ben Koger, and Jake Graving in Constance, with a critically endangered zebra called the Grevy zebra. You could never get permission to do this. So we track them with drones. The drones replace each other when batteries go down. And this is the same resolution I just showed you, but zooming in, using the same software we track the locust and the Drosophila with. Now we can get every time they take a poo, when they're eating, when they're looking, data you could never get from GPS. That gives us much more detailed insight into the social lives of these animals. We're now applying this to elephants as well. And of course, we're taking into account the complex habitat in which the organisms are living. So it's a very, very exciting time to do behavioral studies. It's a revolution. We're making smaller, lighter, cheaper tags. We're able to put out not just tens, but thousands or tens of thousands of tags. This is real data of animals migrating across the planet. We want to understand the global collective to sense the pulse of life on our planet, especially how humans are endangering these critical long-range migrants. These migrants are the first to be hit by human impacts. So how do we get the data back? Well, Martin Mikulski in our institute has developed the Icarus system, which cosmonauts put onto the International Space Station in August last year, and it should be turned on for testing very soon. The space station covers pretty much the entire planet and pings these tags wherever they are to let us know where the animals are. And so this is not using the same Icarus system, but a more conventional system. But see these wing beats? This is real data. We've animated it, of course. But this is real data here. These are storks migrating from Constance in Germany across sub-Saharan Africa. And we can ask how and why they behave the way they do. You may think, how do we know if they're flapping or not? Well, they have accelerometers on them. And when the accelerometer wobbles up and down, that's every wing beat. Then when they glide, this is flat, and then shortly it will start beating its wings again. And we can do this throughout the entire lifetime of these animals using solar power. These are the real trajectories of different birds. And so we can get every wing beat. And then zooming up a level, we can see how they're buffeted by turbulence and eddies. And this is real data where we've reconstructed these thermals. We can ask, how do you decide to leave a thermal, and how do you find new thermals? And we can look at this over the whole lifetime. And we've been able to show, exactly as I'd predicted in the past, that there's a very small proportion of individuals that guide the others on these migrations. And we can predict who is going to guide and who's not using 5 minutes of accelerometry data on the first day of the migration. We can now even predict before they leave the nest who's going to be leaders and not. So it doesn't seem to be genetic, and we've got no idea what's going on because we've only got 32 birds. But now we're tagging 1,000 birds to try to get better understanding. So all of this work is happening in a very beautiful city in southern Germany called Constance. It's on the border of Switzerland, so they literally left the lights on during the war, so it was never bombed. So it's a wonderful old city. My house is 720 years old. This is on Lake Constance. That's Alex Jordan, one of the PIs in our department. It's an incredibly beautiful place to work. Over the lake, you see the Alps. It's really stunning. We're 50 minutes from Zurich Airport, so extremely well connected. It's about an hour to Austria, hour and a half to France, 3 hours to Italy, It's a really beautiful place to live. We have the world's first building for collective behavior. We've got 32 million from the German government to put computer science and biology and psychology and physics together. So not only will we have beautiful views of the lake and the Alps, but underground we have a large animal facility, including an imaging hangar, which is going to be 15 by 15 by 8 meters, allowing us to track in very, very high sub-millimeter precision resolution the motion of animals and to extend our augmented and virtual reality technologies. We're also collaborating with psychologists who are interested in the social behavior of eating. So this is a study that we're doing within one of the facilities of how people are. We also have three-dimensional sound localization. So it's a pretty fun, cool place to be, where we're really trying to push the frontiers of what's possible in behavioral science. We were lucky just to win a 30.5 million grant for 7 years. We can then renew this for the same amount of money for a further 7 if we're successful. So we have many positions available at all levels. We particularly want to establish neuroscience in Constance. And so this is a bit of an experiment, but we're really putting together biology, computer science, economics, psychology, physics, social science, and mathematics. Everyone talks about interdisciplinarity, but here we're actually going to be sharing the same offices, doing the same science, working very, very closely together, and it's an incredibly beautiful place to live and to work. We have the new Max Planck Institute of Animal Behavior, new as of June the 1st, so I don't have any pictures, but we'll have about 80 or 90 million for a new Max Planck building, around 450 researchers focusing on animal behavior broadly, including, again, junior groups and more senior groups. And thanks very much for my sponsors, and thanks very much for listening.
All right, thanks, Iain, for a wonderful talk. If you do have a question, I ask you to speak into the microphone, so I'll hand it to you.
Hi, amazing work. I was wondering, so if you think from a selective point of view about, you could think of several different payoff structures. So one component would be the payoff of everything being coordinated on the same answer. So if you're baboon troop in the predator dictates staying together, right? And then a second kind of payoff is what's the best payoff for everybody, say, in foraging? If we go here, if we go there, okay? And then a third dimension might be different payoffs for different subsets. So I'm just making this up, but the dominants might want to go towards a area where there's less food but more high quality, which they would get. And the others would prefer something else. And so how do these different payoff structures play into the models that you have?
I mean, that's a great question. And you know, with respect to the baboons, it seems that the decisions where to move is incredibly democratic. It could be democratic for two reasons, not mutually exclusive. One is that it has to be because of the uninformed individuals. Just the dynamics mean it becomes democratic. Or it could be that the dominant males advantage from the collective decision-making about where to go, because when they get there, you can clearly see the dominant male can monopolize the resources, right? So there's lots of other things going on in addition. And you also, like with the storks, we can actually measure the costs of different strategies. And you know, we would expect from evolutionary theory that their fitness is more or less the same, but we have this divergence of sort of evolutionary stable strategies. With respect to
the fitness of what to whom?
Of the individuals. So you typically have frequency dependence. You have this branching processes in evolutionary time in terms of behavioral strategy space. And but the case of the fish, I didn't have time to talk about it today, but there we know who's whom, and we can actually give different costs and benefits to different, so we can create these sorts of conflicts regarding the opportunities, regarding the costs and benefits of the outcomes of decisions. And so I didn't have time to talk about that, but that's very much what we're trying to do.
So I don't know if you have data for any system, but is it that the opinionated individuals have stronger information about globally beneficial things, or the opinionated people have stronger opinions about what's good for them?
So again, I haven't shown this and it's unpublished, but we've been working on the rise of the far right within the US. And if you look at everybody, even relatively very conservative people, as, regarding factual information, like what's the state capital of New Jersey or whatever, the more confident people are, the more accurate they will tend to be. There's a strong positive correlation with everybody except the far right. The far right, there's a strong negative correlation, i.e. the more confident they are in their factual opinion, the less likely they are to be correct. And compounded with issues like echo chambers, this then dramatically changes the ability for humans to make good collective decisions. But this is work that we're doing that's experimental work here in the US. So I think there's lots of different ways that we could discuss this because it depends from system to system, but these are very important problems.
Other questions?
Let's see if I got this right. Sort of what I came out with from the first part of the talk is that, which is to me surprising and interesting, is that the fish are individually, they're almost blind in terms of recovering 3D information about the light information and also about predators, because if they're further than a little, so they're almost blind and basically.
Well, they're not blind to predators.
But if they're, you said if they're sort of a certain distance, unless they don't really react too much to them.
Well, if they're occluded from the predator, of course they won't respond to it. So if you're inside the group and there's others in the way between you and the predator, then you can't see it directly.
Okay, so if they're by themselves, then because you said if you disturb the water, they won't react unless they're a certain distance, and it's really they're just really seeing the world. They have specialized mechanisms to see the other fish in the group. And they're basically seeing the world through, they have these specialized mechanisms to detect motion.
Yep.
And it's really through that they can actually sort of make inferences about things that are further away.
Yeah, so of course,
like
if something is close enough that you can see it, if you see a predator directly, then that's great. But you can also see the predator indirectly through responding to the wave of information. And the wave of information that flows through the system is around 10 times faster than the maximum swim speed of predators. So we've been able to show this in the wild using sonar imaging, that predators really struggle with this long-range, very fast collective information processing. And what the predators do is they also form groups. I wonder whether I have it here. Yeah, so the predators also form groups. So we can do these field studies where we track predator-prey interactions, we track their motion, and kind of what's fun about this is the predators really struggle, so they form lines to try and cut the groups apart, to stop them reforming. And it's only when they can cut the groups up into little pieces, whereby the information flow is still fast, but it's very local, can suddenly the predators, real predators, gain advantage over the prey and catch them. Right? This collective information flow is so fast that it's very hard for predators to handle.
I want to pretend for a second that there's a whole slew of children here, and if there were to try to understand this, basically this lecture is about movement. When you have a group of animals together, they're on the move. So let's say if I'm a child, say 8-year, 10-year-old kid, and say, okay, let's say we have a huge flock of birds or fish. Thousands of fish or birds, they're on the move. They're looking for food. That's the top priority. That keeps you alive. So is it one fish or bird that makes the first decision to go a certain direction, or is it instinctive for several individuals to decide that they're going to go a certain direction because they sense food, smell it, or see it in a certain direction?
So typically, there's a low probability of following one individual. You typically will not follow one individual when it changes behavior. But your probability of copying another of individuals, if there are 2 individuals, it's a little bit higher. But once you start getting to 3 or 4 individuals, suddenly it shoots up. And then the probability of that flowing through the system becomes much, much higher. Right? So it's very steep. I showed you those graphs before with the proportion of informed individuals or the number of informed individuals. The probability increases really steeply and then it plateaus. And it turns out that the magic number is around 6 or 7 individuals to strongly influence.
Okay, well, the most famous large groups of animals on the move are the birds and the fish. The giant flocks of birds,
Mhm.
the passenger pigeons, and then the large flocks of schools of herring and whatnot.
Yeah.
So, of course, it's different amongst the different species.
Yeah, you're right. I mean, so we will still predict a similar functional form, but the exact number will depend on other factors. So we can simulate up to, say, I think we simulated 15,000 individuals, and the proportion is very, very small, but you're right, it could be very dependent on the exact system, the critical number that's required.
Okay, so in other words, it's great that you have this, we're doing this research. There's so much to learn.
Yes.
We tapped about 5% of the information, maybe.
Oh, less than 5%.
Okay. I just want to say one thing more before I pass the mic back to you, is that we have endless thousands of students who are studying zoology, studying wild animals, ethology, different levels. And almost millions of students around the world, and billions of dollars spent on research. But all that research is going to amount to nothing if we keep wiping out thousands of species by the hundreds of millions of animals. They're wiping out so many species are in such desperate need. And of course, this research that we're doing is going to help protect them. But before I pass the mic, I just want to say one thing. I hope that there's a few individuals that might come and speak to me after this, because I think it's possible to start a new eco-industry where millions of people spend money every day on food, water, clothing, anything that has to do with money. And instead of people making money, all the profits go to help endangered species and wilderness areas. And I'm working on that, and I need a little assistance to organize that. So I ran out of I ran out of energy. So,
Okay.
but I'm going to stick around after this. Maybe something to talk about after this.
All right, maybe time for one more question. Anybody? We have two more. We'll do you two. And then.
Oh, mine's a simple one. When you were talking earlier about you slow down when you're in the dark place, but you speed up when you're not, you said the curve comes from centrifugal force.
Yeah.
But I didn't understand what you meant, because I didn't
Okay.
know why you just wouldn't just continue in the straight line.
So if you imagine that my hands are connected by an elastic band, is that what you call it in America, an elastic band? A rubber band, thank you very much. I knew it was wrong, but I didn't know how to correct it. So imagine my hands are connected by a rubber band, and that represents the social interactions, and they're moving together, they will tend to move like this. Now if this one slows down, and this one continues fast, that rubber band is going to stretch and stretch and stretch, and start exerting a centrifugal force this way, right?
But what is in the fish, what is the rubber band?
The rubber band is the social interactions, the strength of social interactions. When you become too isolated from another individual, it's very dangerous and you'll lose the benefits of group living. So you're more strongly influenced when you get further away. It's like the rubber band stretching and start exerting a stronger force towards that individual.
So it's not just that one's slowing down in the dark and one's speeding up. There has to be some other piece of information that it's using.
No, it doesn't. If you can do this experiment yourself, you can just take two objects and put a rubber band, and you know, if you yeah. So that's yeah. That's the form of the social interactions. It's like having an invisible rubber band between you
I understand
and the
that
other
that's
individual.
the analogy, but
Yeah,
if it's
but that's actually measurable. This is exactly. Basically it's a breakable spring. So the spring can actually break, but the further away. So if I move away from Jeff, this is going to start exerting a stronger and stronger force.
No, I
which will
understand
pull
that,
me
but
back.
that sounds like it's a second piece of information, not just the being slow in the dark and fast in the light.
This rubber band sort of analogy of the social interactions, it works irrespective of speed. So if you just have two individuals moving at different speeds, then this rubber band will sort of expand. If there's a differential in speeds, then it will cause a bigger gap, which will start exerting a stronger social force. And this is exactly what happens if you measure these properties. within the real group.
I think what Lita's asking is, the analogy is awesome, but she wants to know what's the actual mechanism. It's just a visual
the
signal.
mechanism is visual.
It's a visual signal.
It's visual. Yeah, the
It's a visual
mechanism is
signal,
visual.
motion detect.
Yeah, visual.
That's
They,
right.
they know how to judge distance.
Okay, one more question.
Thanks. So you're showing in the fish example a dependence of group size on the accuracy with which, and then you're also showing animals of various, what we would think of as very different brain capacities following
Yeah.
very simple rules. I'm wondering what predictions your theory makes about ideal group size, or the types of organisms and the types of behaviors that would promote traveling in groups, and the specific sizes of those.
Yeah, I think there's, I think we have to sort of rethink a lot of what we assume with ideal group size. So in the literature, there's something called Sibley group size, which is that you kind of, it costs you too much to get rid of individuals, so groups tend to get much larger than they should do. But many of these ecological theories, these classic ecological theories, don't consider information in any way. So some other work I've done with a guy called Albert Cow is looking at optimal group size, informationally optimal group size. And it turns out, you know, I talked to you about the wisdom of crowds earlier, you know, Sir Francis Galton at the fair. Now, what the wisdom of crowds assumes in all cases is that individuals are making independent estimates based on the same cues. Now, if we think about that for a moment, this doesn't make much sense. So if I'm close to Jeff here, our visual information is highly correlated, and it's very different to yours, right? So correlation of information is going to be more the norm than the exception. And the real world has many different sensory modalities that we can employ, right? So what you can ask yourself, well, what if we break down those two assumptions from the wisdom of crowds? Turns out the wisdom of crowds disappears in 3/4 of the environments, right? And what instead appears is a small, between 2 and around 11 individuals, optimal group size, right? And this optimal group size, we can talk about it later, is really related to the noise inherent in the system, is actually adaptive in allowing the system to escape from over-reliance on correlated information. And so even that, you know, I mean, it's amazing that this whole book's on the wisdom of crowds, and it makes these assumptions which are clearly wrong, but no one has asked what happens if you break it. What about learning, Rescorla-Wagner type learning? You know, you look at the world, there's two options. You have cues, you have a prior of what you're going to expect. You go there, you know, it's maybe tastes bad or it tastes good. You update your learning, fine, right? But if you're now in a group context, you know, I might want to go this way, but we all go this way. So that's decoupled what I wanted to happen from what happened. But I still have to update my learning. And it might not even be clear that Rescorla-Wagner type learning would work. Turns out not only works, it becomes provably optimal. So all those regimes that psychologists love where it fails, no longer fails. So there's all sorts of stuff. And if you think about, you know, taken to the extreme, where these algorithms come from, it's studies of pigeons, rats, humans, they're all social. But people do not study, like I think we're the first group that I know of that's looked at how do rats collectively solve mazes. Yet a single rat in a maze is a classic paradigm, right? So I think we have to really think from the bottom up about, you know, what we're doing when we're just replicating these old studies.
All right, so let's thank Iain again for the information transfer, and enjoy your summer, I guess.